Welcome to BioBits and Bytes. Today we're diving deep into epigenetics, a field that's, well, really changing how we understand our own biology. For you, our listener, who's curious about what makes us tick beyond just our genes, we're going to explore how your genes are actually used. Think of it maybe as going beyond the blueprint to understand the instructions for building and running things. And we've got an expert here to guide us through this fascinating landscape. Our mission today is really to unravel what epigenetics is, how it shapes everything from our development to our health, even how we age. And we'll also touch on some of the, bigger ethical questions that pop up as we learn more. We'll be drawing from quite a range of scientific insights to give you a clear picture. So let's jump right in. Could you maybe start by giving us a clear definition of epigenetics for everyone listening?
Absolutely. Basically, epigenetics is the study of how your cells control the activity of your genes, but crucially, without actually changing the underlying DNA sequence itself.
Okay, so the sequence stays the same.
Exactly. The prefix epi means on top of or above. So you can think of it as like an extra layer of information that sits on your genes. It tells them when and where to be active and, how much.
Right. So genetics is the code, the A's, T's, C's, G's. How is this different then?
Well, genetics focuses entirely on that fixed sequence. Epigenetics, on the other hand, it looks at modifications to the DNA or to the proteins that the DNA wraps around.
Modifications like chemical tags.
Precisely. Little chemical tags or markers. They act like switches, turning genes on or off, or sometimes just dimming them up or down. But the key thing, again, is they don't alter the fundamental ATCG sequence.
So it's like having the same book, but someone's put sticky notes on certain pages saying, read this now, or skip this part.
That's a pretty good analogy, actually, yeah. The DNA is the book, and the epigenome, that's the collection of all these sticky notes, these modifications in a cell, tells the reader which chapters are important right now.
And why is this on-off switch system so vital? What's the point?
Oh, it's absolutely fundamental. Think about it. Every cell in your body, more or less, has the same DNA instruction manual.
Right.
Yet you have skin cells, brain cells, muscle cells, all doing vastly different jobs. How?
Epigenetics.
Exactly. Epigenetics directs this specialization. It ensures a muscle cell turns on the genes for making muscle proteins and keeps the genes for, say, making neurotransmitters switched off.
While the nerve cell does the opposite.
Precisely. It allows for cellular identity and function. Without it, we wouldn't develop properly. We couldn't function.
That makes total sense. Okay, so if these epigenetic marks are doing all this directing, how do they actually work at the molecular level? What are these switches made of?
Good question. There are several key mechanisms, but let's focus on a few main ones. First up is DNA methylation.
Methylation, okay.
This involves attaching a small chemical group, it's called a methyl group, just one carbon and 3 hydrogen atoms, directly onto the DNA molecule itself.
Like a little chemical sticker.
Exactly, a tiny sticker. It often gets stuck onto cytosine bases, one of the DNA letters, especially when that cytosine is next to a guanine base. We call these CPG sites.
CPG sites, and they tend to cluster.
Yes, they often occur in groups called CPG islands, which are frequently located near the start of genes, in the promoter region that controls gene activity.
And putting these methyl stickers there, what does that do? Turn the gene off?
Generally, yes. High levels of methylation in a gene's promoter region usually act as a silencing signal. It makes it harder for the cell's machinery to read that gene, so it effectively turns the gene off.
And removing them turns it back on.
Right. Demethylation typically allows the gene to become active again. So it's a key way cells can lock down genes they don't need long-term.
Okay, DNA methylation, that's one mechanism. What's another major way?
Another really crucial one is histone modification. First, what are histones? Well, our DNA is incredibly long, so it needs to be packaged efficiently inside the cell nucleus.
Right, it wraps around proteins.
Exactly. It wraps around these spool-like proteins called histones. This whole DNA protein complex is called chromatin.
Okay.
Now, histone modification involves adding or removing chemical groups to the histone proteins themselves, not directly to the DNA.
So decorating the spool is not the thread. And how does that affect the gene?
It affects how tightly the DNA thread is wound around the histone spools.
Yep.
Think of it like this. If the DNA is wound really tightly, it's packed away. And the genes in that section are hard to access. They're generally off.
Hidden away.
Yeah. But if the chemical modifications cause the chromatin to loosen up, the DNA becomes more accessible. Then the genes in that region can be read and turned on.
So histone modifications control accessibility. You mentioned different types.
Oh, yes. There's a whole code, really. Yeah. One common one is acetylation adding an acetyl group. This often loosens the chromatin, making genes more active. Think of it like unspooling the thread a bit.
So acetylation generally means on.
Often, yes. It tends to be linked with active gene transcription. Then there's methylation of histones. Now, this is trickier than DNA methylation
How so?
because adding methyl groups to histones can either activate or repress gene activity. It really depends on which specific part of the histone gets methylated and how many methyl groups are added, 1, 2, or 3.
Ah, okay, so it's more nuanced, like different kinds of sticky notes.
Exactly. For instance, methylation at one spot, say H3K4, is usually associated with active genes. But methylation at another spot, like H3K27, is often linked to gene silencing, like shutting down one X chromosome in females.
Wow, that's complex. Are there others besides acetylation and methylation?
Definitely. There's phosphorylation, adding phosphate groups, which is involved in things like DNA repair and cell division signaling. There's ubiquitination, attaching a whole protein called ubiquitin, which plays roles in gene silencing and interacts with other modifications.
So it's like this intricate chemical language written on the chromatin.
It really is.
Yeah.
And these modifications often work together, influencing each other. It's a dynamic system. And we haven't even mentioned RNA-associated silencing yet, where small RNA molecules can interfere with messenger RNA to block protein production.
Okay, maybe we stick with DNA methylation and histone mods for now. So how does all this dynamic control play out during development? You said it's crucial for making different cell types.
Absolutely essential. When you start as a single fertilized egg, all the resulting cells have the same genetic blueprint, but they need to become different neurons, liver cells, skin cells. That's differentiation.
And epigenetics drives that.
Largely, yes.
Right.
As cells divide, specific epigenetic patterns are established. These patterns turn on the genes needed for a particular cell fate and turn off the genes for other fates.
Can you give us that nerve cell versus muscle cell example again?
Sure, a developing nerve cell will use epigenetic marks, maybe demethylating nerve-specific genes and methylating muscle genes, and adding specific histone modifications to activate its nerve program.
And silence the muscle program.
Right, and the muscle cell does the reverse. It silences the nerve genes and activates the muscle genes. It's this precise epigenetic orchestration that builds a complex organism from one starting cell.
Amazing. And you mentioned earlier these patterns aren't fixed forever. They change throughout life.
That's right. While many developmental patterns are quite stable, our epigenome remains somewhat plastic. It continues to change as we grow, as we age, and in response to our environment.
Right. You mentioned that study showing overall DNA methylation tends to decrease as people get older.
Yes, comparing newborns to young adults to very elderly individuals, they found the oldest group tended to have the lowest overall methylation levels. It suggests ongoing shifts in epigenetic regulation across the lifespan.
Which leads us to the environment. Can our lifestyle or diet where we live, can that actually leave epigenetic mark?
Absolutely. There's growing evidence for this. Things like nutrition, exercise, exposure to toxins or pollutants, even psychological stress, they can all cause epigenetic changes.
And these can affect how our genes work.
Yes. And importantly, some of these changes appear to be reversible, which is, quite significant.
Let's talk about nutrition during pregnancy. You mentioned that's a critical window.
It really is. What a mother eats and is exposed to during pregnancy can influence the establishment of epigenetic patterns in the developing fetus. And sometimes these patterns can persist long after birth.
Potentially affecting long-term health?
Potentially, yes.
Oh.
Increasing susceptibility to certain diseases later in life.
That Dutch hunger winter example is so striking here. Could you recap that?
Yes, it was a tragic natural experiment. During that famine near the end of WWII, pregnant women experienced severe nutritional deprivation.
Right.
Decades later, researchers found that the children who were in utero during the famine had higher rates of certain health problems as adults: heart disease, schizophrenia, type 2 diabetes.
Compared to siblings not exposed in the womb.
Exactly. And when they looked at their epigenetics, they found specific differences in DNA methylation patterns, even 60 years later, compared to their unexposed siblings.
Wow. So that early environmental stress left a lasting epigenetic mark.
It strongly suggests that, yes. It highlights how potent early life environment can be for long-term epigenetic programming and health.
What about things we do as adults? Like smoking.
Smoking is another really clear example. Exposure to tobacco smoke causes widespread changes in DNA methylation. There's a specific gene, AHRR, where smokers consistently show lower methylation levels.
But you mentioned this can be reversed.
Yes, Thankfully, studies show that when people quit smoking, the methylation levels at AHRR and some other genes can gradually return towards the levels seen in non-smokers.
That's good news.
It is. It demonstrates the potential plasticity, the reversibility of some environmentally induced epigenetic marks, though how long it takes probably depends on, how much and how long someone smoked.
Okay, so lifestyle and environment matter. Let's link this more directly to disease.
Yeah.
How do errors in epigenetics contribute to health problems?
Well, it's a two-way street, really.
Yeah.
Some diseases can cause epigenetic changes, but increasingly we see that aberrant epigenetic patterns can actually drive disease processes.
Contributing to the cause.
Yes. Think about infections first. Some germs are quite clever.
How so?
They can actually manipulate our epigenetics to help them survive. The example you mentioned was Mycobacterium tuberculosis.
The TB bacteria.
Right. It can cause changes in our immune cells, specifically epigenetic changes that turn off a key immune signaling gene called IL-12B.
Weakening our defenses.
Exactly, making it harder for our immune system to fight off the infection.
Okay, that's infections. What about something like cancer? You hear so much about genetic mutations, but where does epigenetics fit in?
It's a huge player in cancer. Think about tumor suppressor genes like BRCA1, which is linked to breast cancer risk. You can get cancer risk from a mutation in the BRCA1 gene sequence.
Right.
But you can also get increased risk if the BRCA1 gene is perfectly normal sequence-wise. But it gets silenced epigenetically, for example, by heavy DNA methylation in its promoter region.
So the gene is there, but it's switched off inappropriately.
Exactly. So epigenetic silencing of tumor suppressors is a common mechanism in cancer. Also, the overall epigenetic landscape is often profoundly disturbed in cancer cells. There might be less methylation overall, but specific genes, like those tumor suppressors, get hypermethylated.
And different cancers have different patterns.
Yes, often distinct patterns of DNA methylation or histone modification. And this is actually being used now. You mentioned that colorectal cancer screening test, Cologuard.
Yes.
That test works by detecting abnormal DNA methylation patterns associated with colorectal cancer in DNA shed into the stool. It's a direct clinical application of cancer epigenetics.
Fascinating. What about brain diseases? Alzheimer's? Parkinson's?
Epigenetics seems to be involved there too, particularly changes in histone modifications,
Right.
like acetylation and deacetylation, have been observed in brain cells affected by these diseases.
Affecting neuronal function.
Yes, altering the expression of genes important for neuron health and survival. It's an active area of research, trying to understand how these epigenetic changes contribute to neurodegeneration.
And autoimmune diseases too, like lupus.
Yes, conditions like lupus and rheumatoid arthritis also show links to aberrant epigenetics. Abnormal DNA methylation patterns have been found in the immune cells of patients, potentially messing with immune gene regulation and contributing to the autoimmune attack.
It really seems like epigenetics is woven into almost every aspect of health and disease. Which brings us to aging. Is it just wear and tear on the DNA, or is epigenetics a bigger part of the story?
Well, the traditional view focused on DNA damage accumulating. But a newer, very exciting perspective suggests that degradation of the epigenome, the way genes are organized and regulated, might be a primary driver of aging, maybe even more fundamental than mutations themselves.
A loss of the organizational instructions?
Kind of, yes, a loss of epigenetic information over time.
That mouse study you mentioned earlier really highlights this, doesn't it? Can you walk us through that again? Sounds pivotal.
It really was quite remarkable. Researchers developed a system, they called it ICE, for inducible changes to the epigenome to temporarily create non-mutagenic DNA breaks in mice.
Mimicking everyday damage, but sped up.
Exactly. And they found that while the cells repaired the breaks, the epigenetic machinery involved in gene regulation got, well, distracted.
Distracted.
Yeah, the factors that normally control chromatin structure and gene silencing would move to the break site to help with repair. But afterwards, they didn't always return to their original spots.
So things got disorganized.
Precisely. Over time, this led to a scrambling of the epigenetic information. Chromatin became too condensed in some places, too open in others. Genes got turned on or off inappropriately. Cells started to lose their specialized identity.
And the mice?
The mice showed clear signs of accelerated aging. They looked older, their tissues functioned poorly, and their biological age, measured by epigenetic markers like DNA methylation patterns, was significantly older than their actual chronological age.
Wow. So disrupting the epigenome drove aging, but didn't they try to reverse it?
Yes, and this is the most startling part. They used gene therapy to introduce 3 specific genes, Octavor SOX2 and KLF4, often called OSK factors. These are active in stem cells and are known to reprogram cells.
The Yamanaka factors, essentially.
Related, yes. And incredibly, expressing these factors in the aged mice seemed to reverse many of the signs of aging.
Reverse them.
Yes, their tissues and organs regained youthful characteristics. The epigenetic patterns started to look younger again. They essentially performed an epigenetic reset.
That's incredible. It suggests the original youthful information is still there somewhere.
It hints at that possibility, doesn't it? Like maybe cells have a backup copy of their epigenetic software. The study concluded that manipulating the epigenome alone could drive aging forwards and backwards.
Mind-blowing stuff. Which naturally leads to thinking about the implications, including the ethical ones. As we learn more about reading and maybe even writing these epigenetic marks, what issues arise?
Yeah, this is really important territory. As epigenetics moves towards personalized medicine, it brings up a host of ethical, legal, and social considerations. ELSI, they call it.
Like what, for example? Patient autonomy.
Right. How does knowing about your epigenetic profile, which might reflect past exposures or predict future risks, affect your choices? How do we ensure decisions remain truly voluntary?
And privacy must be a huge one. This data reveals a lot.
Definitely. Epigenetic data can potentially reveal information about your lifestyle, your diet, exposures you might not even know about. This raises concerns about data protection, confidentiality, potential discrimination, say, by employers or insurers. And what about unexpected findings?
That leads to responsibility too, doesn't it? If lifestyle affects epigenetics, how do we balance personal choice with public health?
Exactly. There's a risk of blaming individuals for their health based on epigenetic markers that reflect environmental or social factors often beyond their control. This could lead to stigmatization.
Social justice seems key here too. Not everyone has the same exposures or resources.
Precisely. Environmental exposures and lifestyle factors are not equally distributed across populations. We need to ensure that the benefits of clinical epigenetics, like new therapies or diagnostics, are accessible to all, and that we don't exacerbate existing health disparities. On the flip side, epigenetics might offer opportunities for public policy to create healthier environments for everyone.
And this idea of epigenetic determinism, is that a worry?
It is, just as we worried about genetic determinism. The idea that our genes dictate everything we need to be careful not to fall into epigenetic determinism, it's crucial to remember the interplay between biology and social experience. Understanding disease risk requires looking at genetics, epigenetics, and things like exposure to poverty, nutrition, stress, prejudice. It's complex.
Absolutely. It's a lot to think about as the science advances. Just briefly, for listeners curious about how do scientists actually study these epigenetic changes?
Oh, there's a whole toolbox now. Techniques like ChIP-seq help identify where specific proteins, like modified histones, are binding to the DNA. Methylation arrays or sequencing methods let us measure DNA methylation levels across the entire genome. And things like ATAC-seq can map out which regions of chromatin are open and accessible.
So we can actually see these patterns across the genome.
Yes, we're getting increasingly sophisticated maps of the epigenome in different cells and conditions.
Well, that was a truly fascinating deep dive into the world of epigenetics. We've covered so much ground, from the basic mechanisms like methylation and histone modification to its role in development, disease, and even that incredible aging research.
It's a field that's moving incredibly fast, constantly revealing new layers of biological control.
And thank you so much for shedding light on this complex area and breaking down those key scientific ideas for us. For you, the listener, we really hope this has sparked your curiosity. Seeing how factors beyond our fixed DNA sequence play such a critical role, it's quite profound, isn't it?
And the fact that some epigenetic changes can be influenced by lifestyle or environment, and some might even be reversible, offers a really powerful perspective on health.
Absolutely. That potential for change, seen in the smoking cessation studies and even hinted at in the aging research, is really compelling. Of course, the ethical considerations we touched on highlight just how important it is to have thoughtful conversations as this science becomes more integrated into medicine and our lives. This field is constantly evolving. What does it mean for future therapies? Can we develop epigenetic drugs for cancer or neurodegenerative diseases? Could we one day truly intervene in aging? These are huge questions we encourage you to explore further if this has piqued your interest. There are some great resources out there. Thank you for tuning in to BioBits and Bytes.